discuss the development of liver, gallbladder and extrahepatic duct system. First, we will discuss the development of the liver, which is developing during the middle of third week from the endodermal bud arising from the ventral aspect of the gut. So, what is the point? It is the point between the foregut and midgut. So, this is the point where the hepatic bud it appears. Now, this bud grows into the ventral mesogastrium and passes into septum transversum. So, this is the septum transversum. So, these diagrams are basically taken from your standard textbook. Now, bud enlarges and shows a division, that is large cranial part, which is known as the pars hepatica. You can see here. So, this is the large portion going towards the cranial side. Then smaller caudal portion, which is known as the pars cystica. Now, pars hepatica divide into right and left part. Now, right and left lobe of liver, further you can see they are almost of equal size. So, pars hepatica is dividing into these two parts. The right and left division of pars hepatica enlarge and extend into the septum transversum. Next step is the cells arising from the septum transversum are broken into interlacing columns called as trabeculae. Now, next step is the umbilical vein, vitelline vein lying in the septum transversum are broken to form the sinusoid of liver. The sinusoids are also formed from the mesenchyme of septum transversum. So this septum transversum is what? It is a horizontal mesodermal sheath which is lying between the vitalointestinal duct and the pericardial cavity. So we have discussed that location during the development of the cardiovascular system when the folding of the embryo happens and its relation with the heart tubes too. So this liver is developing from that septum transversum. So this septum transversum is contributing to the liver, and we have discussed it is continuing it continue with the you can say or contribute to the development of the diaphragm too. So these main two main components, they are basically derived from the septum transversum. Now, if we have a brief overview of the development of the liver in one slide, so the lobes they develop from the terminal division of pars hepatica of hepatic bud, in which is in coming in contact with the septum transversum. So this portion, that is hepatic bud and septum transversum, they are contributing to the development of the complete liver. How? This hepatic bud is going to form hepatocytes and intrahepatic biliary apparatus, which is the parenchyma, and this septum transversum is going to form the fibrous capsule of Gleason, connective tissue cells, Kupffer cells, hemopoietic tissue, then blood vessels. So, what is the contribution from the vitelline and umbilical veins, which are basically running in the septum transversum? As we have discussed that these veins, they break into small channels or trabeculae or columns. So, they are going to form the sinusoids. So, this is a brief overview of what we have discussed. Now, coming to this diagram, this is the histological picture of the liver. So we can see the hexagonal picture of the hepatic lobule, in which the hepatocytes, they are present in a radiating manner starting from the central portion going to the periphery. Now we have discussed that is these hepatic sinusoids which are present in between these hepatocytes. Here you can see here, so they are derived from the vitelline and umbilical veins. The hemopoietic tissue in fetal life is derived from the septum transversum. So the bile ductules and bile canaliculi, they are derived from the hepatic bud. So this is the arrangement or histological arrangement of the liver, in which the portal triad is present at the angles containing portal vein, bile ductules and hepatic artery. Now it is clear that bile ductules and bile canaliculi, they are developing from the hepatic bud, which was appearing from the junctional point of foregut and midgut, and the breaking of these vitelline and umbilical vein is going to form the sinusoids of the liver, and septum transversum is going to form the hemopoietic tissue in fetal life. Now, what are the different anomalies of liver? The rudimentary left lobe, then anomalous lobulation, then absence of the quadrate lobe, then Riddle's lobe. It is a tongue-shaped projection which is coming from the inferior margin of the right lobe of the liver. This is the Riddle's lobe, or this you can see in the diagram too, Riddle's lobe. So next is accessory ligament falciform ligament. So the liver is developing in this septum transversum portion, and you can see it is present in the ventral mesogastrium, and part of the tissue is separated from the main tissue. It is going to develop into accessory liver, independent liver tissue. Then coming to the gallbladder and extrahepatic biliary apparatus. Gallbladder and extrahepatic duct system. In this, the gallbladder, it develops from the pars cystica. We have discussed the one bud, first hepatic bud, it appears, it is dividing into cranial portion, that is pars hepatica, and caudal portion, pars cystica. Now this is the pars cystica which is going to form the gallbladder and cystic duct. Now bile duct arise from the hepatic duct proximal to the pars cystica. Now coming to this, initially bile duct open on the ventral aspect of the duodenum, but as the rotation of the gut and differential growth of duodenum is happening, it comes to open on dorsomedial aspect of the duodenum along with pancreatic duct. Now keeping in mind this development, what are the different anomalies of gallbladder? First we will discuss the anomalies of shape. In this we will discuss the Phrygian cap and Hartmann's pouch. Phrygian cap is basically fundus may be folded on itself to form a cap-like structure, and Hartmann's pouch is just projecting from the wall of the infundibulum downward as a pouch which may be adherent to the cystic duct or even to the bile duct. So this is Hartmann's pouch and this is Phrygian cap. Now anomalies of position. The transverse position on the undersurface of the right lobe or left lobe, you can see here. So this is the gallbladder is lying here. The position may be horizontal. Then floating gallbladder is enclosed by peritoneum on all the sides. The gallbladder is enclosed by peritoneum on all the sides. It may be attached to the liver by fold of peritoneum, or it may be completely free. We call it as floating gallbladder. Then intrahepatic gallbladder is, it may be embedded in the tissue of liver. So normally it is present on the inferior surface of the liver, so it is present in a direction like this, but if it is placed horizontally, so that again become the anomaly. If it is surrounded all around by the peritoneum, we call it as floating gallbladder. Now sometimes the lumen of the gallbladder is completely or partially divided by a septum. This is a partial septation of the gallbladder. This is the complete. Sometimes what is happening, even it is reaching up to the cystic duct too. So this is known as the duplication of gallbladder. But in this case, in diagram B, you can see the septation of the gallbladder cavity, but the cystic duct is common. But here the division of the cystic duct is too. So it is opening with into the main hepatic duct, that is by the two openings. You can see here independently these two openings, they are opening into that duct, but here the single opening is there. So these are the different anomalies, or when the arrest just occur at different levels of the development, this anomaly it appears. Then sessile gallbladder directly open into the bile duct instead of cystic duct. So this is sessile gallbladder, no cystic duct is there. Then agenesis of gallbladder is another anomaly which can happen. Agenesis means non-development of the gallbladder. Then diverticula may arise from any part of the gallbladder, like Hartmann's pouch. This is sessile gallbladder. Now coming to the anomalies of the extrahepatic duct system. This is the normal. Now here we can see the right and left hepatic ducts join within the liver substance. So right blue colored, right and left, they are just joining within the liver substance. There may be the long hepatic ducts may be there. Then very long cystic duct may be there. You can see here red colored. Then is normal, if we see the opening of the cystic duct into this portion. So cystic duct passes behind the common hepatic duct and join its left side. Normally it is just joining the common hepatic duct like this. But if it is rotating and coming on this side, then it is joining on its left side. Sometimes this rotation which is just happening, it may pass posterior to the hepatic duct, or it may pass in front of the common hepatic duct and join on left side. Or this cystic duct may open into one of the two hepatic ducts. So that may be the another anomaly which can happen. This is the anomalies of extrahepatic duct system again. So there may be the complete agenesis. There may be agenesis of the gallbladder and cystic duct is not developed. only cystic duct may be missing, then hepatic ducts may be missing, then bile duct missing. So these are the anomalies. Sometimes the some portion of the terminal bile duct is missing. So these are the anomalies which are happening during, may happen during the development of the extrahepatic duct system. So this process is known as agenesis. So this is the next anomaly related with it. Now coming to the anomaly of the extrahepatic duct system, further what can happen? So we have discussed the agenesis, we have discussed the duplication. So this is the duplication of the duct system now. So in this, what is happening? The cystic common bile duct, it may be separated at the you can say at the point of its termination, near it the point of the termination. So this may be leading up to the opening of the, or it is just reaching up to the level of the cystic duct, where the cystic duct is joining the common hepatic duct. So this duct, common bile duct, may be divided at this level, or there may be the complete system starting from the hepatic duct, coming to the common hepatic duct, then cystic duct. So independent duct system is developing. So this is the another anomaly of the extrahepatic duct system, and we call it as duplication. So thank you very much. In next class we will discuss the pancreas and spleen. If you like my videos, you like my classes, kindly share it, subscribe it, and like it. Thank you very much.